So for my project, I decided to do CRISPR being used for sickle cell disease. To review what sickle cell disease is, it's a mutation in the hemoglobin cell, which causes the structure of the red blood cells to form into a hard sickle shape, and this causes many complications, for example, blood vessels clotting, chronic organ damage, and frequent pain throughout the body. Most treatments are only to relieve the symptoms, but aren't able to cure the disease completely. The one cure that they did find was the stem cell transplant, which is not very common because you have to find a matching donor. And this is just a comparison of a red blood cell, a normal one compared to a sickled one. And as you can see, the normal one is rounded, while the sickled one is very sharp, which makes it hard for it to be able to flow within the blood vessels, which gets it caught, and that's what causes the clotting. Sickle cell is a genetically inherited disease, which makes it a prime candidate for the gene editing tool. And there are a couple ways that the CRISPR could be used, but they've only used one on humans. Before the trials had begun on humans, it had been tested on mice. And the one trial that they did do involved fetal hemoglobin, which is a protein, and it is produced during pregnancy and a little bit after. And the scientists take the gene out and edit it, and they reinfuse it back into the body, which makes the patient be able to produce fetal hemoglobin. And the point of doing this is for the protein to compensate for the defect in the hemoglobin cells. And the first patient to undergo the process of getting their genes modified has shown great improvements, and the patient's protein levels have been elevated to the point of no longer having chronic pain. This is just one of the methods that they've used, this is one of the methods that they've talked about, which you would assume that they would just edit the hemoglobin gene, which causes sickle cell, but that's way more complicated, and that's why they haven't taken that road. And some of the overview benefits would be that it alleviates the pain, and hopefully in the future they're able to actually cure the sickle cell disease completely, which I would say is pretty beneficial. There is a large amount of people suffering from this disease, and very few are able to live a full life, so for it to be cured would be great. It's not only just being used for sickle cell, it's also being used for other diseases of the blood, as well as they're talking about, and I'm sure they're slightly experimenting with cancer as well. Some of the only ethical implication debated that I could find had to do with gene editing being used to make a long-lasting heritable change at the embryo stage or on germ cells. But in my opinion, it shouldn't be ethically debated just for the fact that it's curing someone of a painful disease. But in the end, will always be an ethical debate on gene editing revolving around the creation of a child. But yeah, here's my references. But yeah, that's my presentation. Thank you.